Hi, everyone. You saw me a little while ago, but to reintroduce myself, I'm Tim Ogden, the president of the Bardet-Biedl Syndrome Foundation. And today on this video, I'm joined by Dr. Max Nachury. Any of you who visited our last in-person conference here from Salt Lake City back in 2018 will remember Max. He is a researcher who's been studying a lot of the molecular basis, the protein basis of BBS. And so I'm going to be asking him some questions to help him bring us up to speed on the research that he's been doing. Max, thank you so much for joining us. But why don't we just start with, there are a lot of people who would not have been in Salt Lake City. Would you introduce yourself, your lab, the people that you work with, and what you study?
Absolutely. Well, Tim, it's a pleasure to talk to you today. So my name is Max Nachury. I'm a professor in the Department of Ophthalmology at University of California, San Francisco. I've had my lab now since 2008, first 9 years at Stanford, and then I moved here to UCSF. And I've been fascinated with Bardet-Biedl syndrome in particular, and with diseases of the cilium, the so-called ciliopathies in general, pretty much for the past 15 years. And it's really something that's just kept me ticking for all this time because of the basic biology that is so interesting, and because of, I'm hoping, the contributions that I can make to these diseases and to the life of the patients.
Thanks. Now, Max, you focused especially on what's called the BBSome, and most people are familiar that there are different BBS genes, but can you explain what the BBSome is and how that relates to the BBS genes?
Absolutely. Yeah, so this really goes back to the very beginning of my investigations on BBS. And this was 2005 when I started to hear about this enigmatic disease named Bardet-Biedl syndrome. There were just a handful of papers on it at the time, and the first few genes had started to be identified. So clinical geneticists had done mapping of pedigrees to get to critical interval and identified the causative genes for BBS. Had identified BBS1, BBS4, and had started to make some links to the biology of the cellular organelle named the primary cilium. So I'm going to go to the board and draw a few things for you here. So if you think of any given cell in your body, I'm just going to draw that. That would be a typical cell where you have the nucleus, the repository of genetic information, so where all your genes are located. And what people started to appreciate circa 2003, 2005 is that there is this structure here that's called the primary cilium. And pretty much when I started to read and hear about research on BBS at the time, all that was known was the identity of these gene products. So these proteins, so all these genes that are defective in BBS patients, they are expressed to give rise to individual proteins. And proteins are really what function in the cells. That's how you transport molecules across membranes. That's how you can modify the property of membranes. That's how you can have enzymes that are going to be important to make specific chemical compounds, and so on and so forth. I mean, the proteins are really the tiny little engines of life, if you want, and this is really where the biological defects are going to come out. So anyway, these BBS proteins that had been discovered, we still didn't know what they were doing in the cell, and that's what I started to get interested in. So what I did when I was a postdoc back then, in 2006, 2007, was to ask a very simple question. Of these proteins, what are their friends? Proteins rarely function as individuals, they function together. It's like in an orchestra, you're going to have different parts of the orchestra that need to play together in order for the symphony to be properly executed. Well, I just wanted to ask this question, what's in the BBS orchestra there? And that led to the discovery of this protein complex of 8 BBS proteins that I call the BBSome. And this BBSome consists of BBS 1, 2, 4, 8, 7, 9, and a genes that we later discovered as BBS 18. So this complex, then we had discovered it. As I said, this is now we identified the players in the orchestra, but we still were missing what the symphonies that they were playing. And that was the very difficult question to figure out what cellular process do these proteins participate in. So that took a long time because this is a game of trial and error, if you want, where you formulate hypotheses, you use educated guesses from first principles, from the literature, from what does the protein look like, and you start to posit various hypotheses, which then you can test in these biological systems. But over the years, we have arrived at what I think is a fairly well accepted model for what the BBSome does. And it is as follows. So what we understand now is that in the cilium, you have a variety of what we call signaling receptors. So you have signals that come from the outside of the cell that are received on the surface of the cilium. Okay, you just have from the environment of the cell some messages that arrive, and they're interpreted at the surface of the cilium by what I'm drawing here as a circle. That's our signaling receptor. Well, these signaling receptors, you really need to have a high level of coordination of the signaling receptors that's in the cilium versus the signaling receptor that is out of the cilium. And it's a very fine balance here that the cells have to reach in order to interpret these signals from the outside. Too much, too little of the receptor in the cilium, and now the information is not decoded properly. And here is where the BBSome plays its essential function, which is that it takes a signaling receptor that is in the cilium and it takes it out of the cilium. And this very simple task has, in many contexts that we've studied, great biological consequences in that you can downregulate, you can dampen, if you want, the signaling level of a specific pathway, or in some cases, you can also increase now with a more complex loop. You can sometimes increase the activity of a given signaling pathway. So there you have it in a nutshell. This is really what the BBSome does, and really the cellular function that underlies, we believe, all the defects in BBS patients is a failure to remove signaling receptors or other molecules from the primary cilia.
So the idea that BBS comes from this basic signaling defect, that messages aren't being passed, helps make sense of a lot of BBS symptoms, things like limited sense of smell, retinal degeneration, some of the kidney disease. But it's also a little curious that it feels like nothing in your body should work if signaling doesn't work. So why is it that in BBS, it seems like some signals don't work, but some signals do?
That's a great question, Tim. So in fact, we know from studies in mouse models that if you make an organism without a cilium, it cannot execute the proper developmental plan. Okay, you cannot make a newborn mice without a cilium. You cannot arrive at this stage. So therefore, why is it that you can alter the function of the cilium in BBS and yet be compatible with development? I think that's a fascinating question here. The answer I would have is severalfold. So one of them is that what the defect is in BBS is an imbalance in the level of signaling molecule and other molecules in the cell. So you have too much of these red molecule in the cilium in BBS patient cells. That's not quite as detrimental to the interpretation of extracellular signal as not having any cilium at all. So that's the first part of the answer. The second part is that what we've discovered is that molecules can go out this way, the way that I drew with this red arrow there, going back into the cell. But a few years back, we discovered that molecules can also get out of the cilium. Another way is that they can just get ejected from the cilium into the extracellular environment. And we view that pathway, this second non-conventional pathway for exit from the cilium, as a safety valve, if you want, for the cilium to get rid of excess material. And so that might allow the cell to cope with defects that arise from loss of BBS function.
Now, you articulated the proteins, the genes, and the proteins they code for. Clearly, I need to turn on a light here in my office in the BBSome, but of course, there are a bunch of other BBS genes as well. What do those have in common? Do those proteins also interact with the BBSome in some way, or are they doing something completely different?
Yeah, and I'm sure that's a very important question to a lot of families who have patients who don't have genes in any of these BBSome subunits. And I do now realize that I forgot one of them, which is BBS5. But we have, give or take, 21 BBS genes that are known. There are probably others that aren't known yet. But for example, BBS10 is a gene that's frequently mutated in the BBS population. It's not a subunit of the BBSome. So how does BBS10 deficiency connect to the BBSome? And there, this is not our work, but the work from Val Sheffield has shown that, and again, going back to this notion of protein complexes, little orchestra, quartet, whatever musical ensemble you want, that need to play together. Well, in this case, what you have, 3 genes, BBS6, 10, and 12, and they assemble into a protein complex whose job is to assemble the BBSome. So there you have at least the explanation for why loss of either BBS6, 10, or 12 function will secondarily lead to the loss of BBS function, because this BBSome can no longer properly assemble in the absence of BBS6, 10, 12. And I would say that by now, most of these 21 BBS genes that are known, we understand their function in terms of either interacting with the BBSome for the purpose of removing proteins from cilia, or in regulating the assembly, the folding of the BBSome. So I would say at this point, we can very clearly put the BBSome, for example, with BBS3, both of them function very close together at this step. We have other proteins such as BBS17 also, that is very closely physically associated with the BBSome. BBS19 that we have found to regulate the activity of BBS3. Yeah, I could go on and on, but this very much now we have, I would say we have hit a point of convergence for all these different BBS gene products, that is the BBSome.
So there's a level of curiosity here of really understanding, okay, why is this happening at the cellular level? Why is it important to understand what the proteins do, what the BBSome does, in terms of pointing the path to future therapy?
Yes, and you're absolutely right. We need to justify what we're doing, and at some point, it becomes important. And I do know that all of the people who are listening here, this is what they want to know. So I would say at this point, we still don't understand. And I'm just going to take one example, which is loss of vision. Okay. We don't understand how the accumulation of protein in the cilium of the photoreceptor cells, and so I'm just going to draw that for you here. So I'm going to draw the photoreceptor cell for you. That's a cell here. This is a cell that senses light, okay? And there, I'm just circling the structure that corresponds in this one cell to the cilia. This is the cell type that degenerates in BBS patients. We do not understand why having an excess of these red circles, which represent the factors that the BBSome is taking out of the cilium, why this leads to the degeneration of this cell type. But this is now work that's ongoing in my lab, where we are trying to identify the nature of these red circles. just knowing what are the proteins that the BBSome is taking out of the cilium. And I think what is really promising in this type of research is that we are hoping to now understand the biological pathways that goes from BBSome is not functional, these red factors accumulate in the cilium, to the next step, which is these cells undergo degeneration. And I believe that if we're able to understand this pathway, and again, I'm just talking in the case of photoreceptors and retinal degeneration, but the hope is that if we can understand the mechanics that go from you lose the BBSome, you accumulate protein in the cilium, this cell type undergoes degeneration, we might be able to have to design therapies there that could interfere with the degeneration of these cell types.
So, Max, I know that this is sort of a good transition to the fact that there's still a lot that we don't understand. And I know that you are not the kind of person who said, oh, BBSome, we got it, I'm done, kick up my feet, my work here is done, I'm retired. I'm sure you have a lot of questions. So I'm really interested in what you think, what questions really interest you. Where do you think we go from here? What's the most important things for research to uncover?
Absolutely. So I would say one question is the one I just told you about. We're really interested, and now I have a couple of people in my lab who are working with mouse model of BBS, who are purifying cilia from photoreceptor cells, who really want to understand what is a molecular defect here that is leading to photoreceptor degeneration. That's now a major project that's going on in the lab. The other aspect that fascinates us is obesity. A group at UCSF made a very important discovery in that and I was just drawing you here some receptors that were on the surface of the cilium, and I was telling you earlier how important they were to decode signals from the outside. Well, it turns out that there is one such signal in the body that limits your appetite. Okay, that signal is interpreted, in fact, on the surface of the cilium by a receptor named melanocortin receptor 4, or MC4R. Well, this MC4R receptor, and it was only a few years ago that we learned about that. This is, in fact, a colleague of mine here at UCSF, Christian Wess, whose lab discovered that MC4R needs to be at the primary cilium in order to sense these signals from inside the body that limits your appetite. Okay? But we still don't understand exactly why a defect in the movement of MC4R outside of the cilium would lead to less MC4R dependent signaling. This is still something that's very enigmatic, because we know that MC4R needs to be on the cilium in order to sense these cues that the body sends to say, okay, let's stop eating. But why is it that having even more MC4R on the cilium should be detrimental to this pathway? There is something of a disconnect here that makes it very interesting. For us, what interests us most is not the things that we understand, but the things that we don't. And there, it's really an enigma at this point about why this movement that the BBSome carries out, this movement of taking a receptor out of the cilium, why it should lead to a defect in this MC4R dependent limit of appetite. And so that's really what I would say the two axes I told you about are what we're interested in now.
Well, I mean, I love that example, Max, because as I'm sure you're aware, Rhythm's treatment for hunger and boosting the MC4R pathway was just approved a few weeks ago. And you know, that was an important step of figuring out that there is a pathway here that sort of leads to the possibility of some research there, but there's a whole lot we still don't understand that could yield lots of other possible treatments for not only the obesity, but vision loss, and along the way, potentially some of the other symptoms of BBS. And so on behalf of the BBS community, let me say thank you to you and your lab. Those folks you just talked about purifying the cilia. I know that's a very painstaking work that takes a very long time to do, but critically important to all of us who are someone with BBS or love someone with BBS to know that there are people like you and the folks in your lab who are working every day to try and illuminate, help us better understand the syndrome and illustrate the pathways that we can pursue that we hope ultimately lead to treatments, and why we're all watching this conference is in the hope that we eventually get there. So let me say thank you once again, Max, for your time. And
thank you, Tim.
We will look forward to seeing you at a conference in the near future. Hopefully we'll maybe even be able to have a regional gathering in California there in San Francisco sometime soon.
That would be wonderful. Thank you, Tim.